Think of the Valentine's Day and present your partner with this. The signet ring. Today's class is on the signet ring cell lymphoma. A weird and strange topic, unfortunately, in our question papers. Whenever we get a topic like this, it is mandatory that we should be knowing these subheadings. Think of the subheading will take you to the answer. Seemingly we might be knowing it, but when it comes to answering, it is difficult. What is the introduction? The origin of this word, morphology, the markers, neoplastic and non-neoplastic signet ring cells. Can they be found in the body fluids? What is the differential diagnosis? Other methods of diagnosis and the pitfalls. And what I'm showing you on the screen is a signet ring. Those days, it used to be the ring of a king with which he would endorse his emblem. That is the signature. So I find that it is hollow in the center and thickened at one end, which is exactly the morphology of a signet ring. Now that the topic is a signet ring cell lymphoma, lymphoma by itself is a nightmare for all of us. I will not be going into the details, but it is prudent that we know the development of this. Lymphomas are basically of two types, the B cells and the T cells. When it comes to the B cell, you find that there can be a naive B cell arising from the stem cell. And within the node, you find that there can be a mantle zone. Nowadays, you are getting terminologies such as the mantle zone lymphoma, etc. And there is a light zone and a dark zone. You find that there are a series of cells, such as the immature cell or the precursor cell, and then the immunoblast, etc. Those days, the classification used to be large cleaved, large uncleaved, and then small cleaved, small uncleaved, etc. So I find that at each of this, you find a typical type of a lymphoma can originate. For example, a mantle zone lymphoma, Burkitt's lymphoma, all this can lead to a follicular lymphoma. And finally, you find that the B cell matures, one of which goes in for memory, the other matures into a plasma cell and antibodies, for which function the lymph node is there. So this is, and this table gives you a beautiful origin of the various types of lymphomas. Have it in one corner of your mind. The term signet ring cell lymphoma was introduced by Kim et al. In 1978. He had seen about 7 cases of non-Hodgkin's lymphoma which were showing this morphology. At that time of presentation, there were about 50 cases, and now more than several hundred cases would have been found. The lymph nodes are strongly positive for this lymphomas, unlike elsewhere. Bone marrow is very uncommon. It can be associated with a follicular lymphoma, and very occasionally there can be a T cell signet ring cell lymphoma. Again, the signet ring wherein I am finding the emblem and the hollow. The general features occurs in the age 20 to 80 years, most common is around 50 years of age. Location in the serosal region of the GIT, presacral, peripancreatic, retroperitoneal or retropancreatic, para-aortic lymph nodes. Urinary bladder is another location. It is generally a circumscribed large lesion, which is usually solid or can be solid and partly cystic. And one thing that is important is the plane between the mass and the surrounding fat is well maintained. In the sense, it is not infiltrating. There is one entity called the fluorodeoxyglucose positron emitting tomography. So that is used for the diagnosis. As I told you, there are other locations where you people can see, and strangely, this is a picture courtesy web pathology wherein I am finding Signet ring cells in the breast. The salient features again of a Signet ring cell neoplasm, in this case lymphoma, is a clear cytoplasm which we are seeing. Excess production of immunoglobulin. After all, its basic function is to produce immunoglobulins. There can be eosinophilic globules in the cytoplasm, which can very well be mistaken for mucin. PAS is again adding to the O's positive. It can express IgM, and the clinical behavior is similar to that of a follicular lymphoma. So this is a histopathology, and this is a slide of it. I shall be coming to the description of it later. Thanks to nature. Aggressive nature of the B-cell lymphoma is shown in this slide. I mentioned the differential diagnosis. Lezoni and Mills have compiled the entire list of differential diagnoses. So, in that one, I find that there can be a Signet ring cell adenocarcinoma in the stomach, GIT, biliary tract can be mistaken, prostate, bladder, endometrium, endocervix, salivary gland, and whatnot. Maltoma, carcinoid of the gallbladder, GIST, schwannoma. All these are neoplastic lesions which may show Signet ring cells. Again, there is a list of non-neoplastic lesions such as inflammatory conditions, pseudomembranous colitis, ulcerative disease, not colitis, therapeutic intervention by means of surgery or chemotherapy, and erosive cholangitis. So, an IHC is mandatory for the confirmation of a diagnosis. Malignant Signet ring cells in the pleural fluid. They have a larger hyperchromatic nuclei, irregularly condensed chromatin, peripherally located, and on one side the nucleus is flat, not indented. There can be numerous cytoplasmic small vacuoles. Nucleoli can be present, and subtle changes can be mistaken for the mesothelial cell. Remember, this is pleural fluid, and histiocytes with degenerative changes. That is what is shown in this picture, and these are some kind of having a flattened surface, probably malignant. And mesothelial cell versus a lymphoma, and also an adenocarcinoma. This particular table, I would like you people to kindly memorize the difference between a mesothelial cell and an adenocarcinoma. Usually we have got this problem. So the carcinoma in this case, it is not a lymphoma, numerous vacuoles. You find that the vacuoles can be small, border of the vacuole is ill-defined, and nuclear size is large. This is a catch over here. And on one side it is flat, there can be clusters of cells, not a single cell. Thanks to this reference. And the laboratory diagnosis can be identifying the atypical lymphoid cells, in our case, which is diffusely immunopositive for CD20, 79A, CD10, etc., and negative for CD3, CD5, CD30, and MUM1. Epithelial pancytokeratin is again negative because it is not an adenocarcinoma. Mesenchymal desmin is negative, SNRAT is positive in adipocytes, which can again mimic this. And you find that BCL2 gene immunofluorescence can be done. Musicarmin is negative, and it is positive in the case of epithelial cells. There is no bone marrow involved. It is a particular case where there is a peripancreatic biopsy, and it is a lymphoma with clear Signet ring cells that you are seeing. And there is also an adenocarcinoma of the pancreas to add to the confusion. The salient features to the pathologist. Now, the Signet ring cell lymphoma is highly infrequent. It is deceptive. Epithelial, mesenchymal, hematolymphoid neoplasms can mimic these. They are more common, however. The management is different. That is why the final diagnosis matters. Extranodal sites can be the serosa and sometimes it can be misdiagnosed because of an infiltrating adenocarcinoma. The lymphoma is usually nodular, not diffuse, and it has got angulated and cleaved nuclei. Whereas in the epithelial, it is diffuse, commonly observed in carcinoma, and also the antibody and molecular diagnosis will be useful. So this is one particular case where there is a large mesenteric lymph node in a 36-year-old male, the same reference as I had quoted earlier, and it was not diagnostic but suspicious. And when it turned out to the histopathology, it found out that there was a Signet ring cell follicular lymphoma. So this is another case from Nature which I had shown you people earlier. Thanks to your listening, and this is the Signet ring.